Okay, students, good morning. This is Mr. Emir speaking. So pretty sure you guys have done the quiz in Kahoot just now. I just want to recap on what you guys have learned in the quiz. Okay, so these are the structures that it is compulsory for you to know before you proceed with meiosis. Okay, so this is a very much conceptual topic, so you should know all of these structures for you to understand um, the whole of chapter 6. Okay, so in the first picture we have two chromosomes. Okay, they are homologous because these two have the same set of genes. What this means is if you look here, so if we uncoil this structure right here and we get a chromatin, okay, one section of the DNA is a gene. Okay, what this means, if this section of DNA, if you transcribe it, you can get a protein, a specific protein to this gene. So for example, if this gene codes for the color of your eye, okay, so if you coil it into a chromosome, okay, this gene will be in this chromosome, okay, it will also be inside this chromosome, okay, the gene for the color of your eye is also going to be in this chromosome. That's why these two, we call them as homologous chromosomes. So if we put this shaded region here, let's say we label in this region, okay, there's also gonna be a region here. This is also a gene for the color of your eye. So another way to picture this in your head is if we I'll show you one example. So imagine this thing is a strand of DNA. So we have a very long piece of DNA. Okay, so this is all one piece of DNA. Okay, one piece. So very long, we call it chromatin. Now, if you try to wrap them around your hand like this, oops, wrap it. Wrap and wrap. Okay, so imagine your DNA is coiled around your hand like this. So this whole thing is a chromosome. And each part corresponds to a specific gene. So for example, the gene right here codes the color of your eye. This gene right here codes the color of your hair. And this gene right here codes the color of your skin. So every chromosome has these regions that codes for specific protein and codes for specific characteristics. So this particular chromosome right here codes for the color of your eye, color of your hair, and the color of your skin. Okay? Now, one very important thing to note is, even though these two are homologous, which means they have the same gene at the same place, it doesn't necessarily mean that they have exactly the same base. So in a DNA we have, so example here we have A, T, G, C. This one is C, G, A, T. Okay. Now, this might be true to this chromosome, But this one, maybe it has A, G, C, T, T, C, G. Okay, you notice there's one difference right here, but this region, okay, is the gene that codes for the color of your eye. So this difference might correspond to what is the color of your eye. So probably if you have this sequence, you have brown eye, and probably if you have this one, you have green eye. Okay, so they're not identical, but they are homologous, which means they have section of DNA that code for the same characteristics. In this case, it is the color of your eye. Moving on to the next picture, we have these two right here. Okay, now these two are very similar, but for this part, one chromosome actually duplicates into two identical parts. Okay, remember in cell cycle, in interphase, we have S phase. That is the part when one chromosome, chromatin actually, the chromatin will replicate itself into two. So over here we have one chromosome or one chromatin, actually very long one. Okay, when it replicates, it becomes two. So the question is, how many chromosomes are here and how many chromosomes are here? So this one has two chromosomes. This one is still two chromosomes, but for one chromosome has two chromatids. Okay, so one chromatid, two chromatid, one chromatid, two chromatid. Okay, so in total, in this picture, we have 4 chromatids. Now, in one chromosome, okay, we have 2 chromatids. Now, because they are completely identical, remember, they replicate from 1 into 2. So these 2 parts are identical. These 2 are also identical. So this one and this one are sister chromatids. This one and this one are also sister chromatids. On the other hand, if this one and this one, because it's on a different chromosome, okay, we call them non-sister chromatids. Okay, this one and this one is also non-sister or non-identical actually. So this one and this one is non-identical. This one and this one is non-identical. This one and this one is also non-identical. Okay, different colors, different chromosomes, non-sister or non-identical chromatids. Okay, in meiosis, which you will learn after this, okay, there's gonna be a special event that does not happen in mitosis, which is the crossing over. Okay, now crossing over, or in other words, recombination, they happen when you have 2 homologous chromosomes. Okay, one of the chromatids from each chromosome overlap. Okay, like this. Okay, this region we call it the chiasma or chiasma or whatever you want to pronounce it. Okay, and the overlapping of these 2 non-identical chromatids, we call it as a synapsis. Okay, so synapsis. Okay. Okay, by the way, don't forget, okay, this whole structure right here in this picture right here, okay, we call it a bivalent, or a tetrad. Okay, the reason why we call it tetrad is because we have 4 chromatids. Okay, 4 corresponds to tetra, so tetrad. Okay, bivalent is because we have 2 chromatids that are covalent or like bonding to each other, we call it, so bi is for two, so bivalent. Okay, so you can call this whole structure a bivalent or a tetrad. Okay, now when the synapses happens, when those two regions overlap, now what's gonna happen is the part where it overlaps is going to exchange. Okay, so the blue part on the paternal chromosome is gonna exchange parts with the red part right there. So this process is called crossing over or recombination. And as a result, you can see here, the paternal chromosome has one piece of the maternal chromosome part right there. Okay, and the maternal has a piece of the paternal chromosome right here. So this does not change the characteristic of the genes. It's just that it will form new combination of genes. It's hard to describe this right now, but just so you know that this process is very important to to give variation to a species of animal or plant. Okay, so if we don't have this, then there's no variation in our species. Everyone will look the same, no difference at all. This is what makes meiosis very unique.